Today, today we're going to move on to talking about transportation within cells, and we're going to start off with diffusion. Diffusion is the movement or motion of all liquids and gases from areas of high concentration to areas of low concentration. Eventually, the particles will be equal spaces away. This is very much like I gave the analogy the other day in class of the freshman girl that sprays on perfume at the end of the hall, and by the end of the hour, or even by the end of passing period, we can smell the perfume all the way down to the other side of the hall because the perfume particles have diffused across the hallway. It's, you can think about just anything, perfume, baking cookies, all that stuff. When you can smell it, it's diffusing. If you've added dye to the water, you can see the dye diffuse through the water. It's just the movement of particles from high concentration to areas of low concentration. Moving on from diffusion, we have osmosis, and osmosis is the movement of water. Okay, so osmosis is the movement of water. Diffusion was the movement of particles. Osmosis is the movement of water from areas of high or areas of low solute concentration to areas of high solute concentration. There is a video link that I will add. I don't think I can bring it up right now. And I'll add it also to Sophia, and I'll talk about how osmosis works. Osmosis is important, especially for our blood cells and all our cells of the body, because if we have too much solute outside of our blood cells or cells in general, all the water from our cells will go into the liquid that surrounds our cells because there's more solute over here, and that's where the water wants to be. In the end, you can see that our blood cells here end up being very shriveled up because all the water has left them. In the middle, we have isotonic, okay, and that you can see that we have water going out and water going in. This is kind of the happy medium. We have the same amount of water going in as we have water going out. And at the far end, we have hypotonic, okay, hypo. We have way too much water coming in because the inside of the cell has more stuff or more solute, and that's where the water wants to be. You can see the blood cells, red blood cells down here, and they become very full. And actually, if you Google some of this stuff or YouTube it, you'll actually find microscopic views of the red blood cells bursting because they have too much water in them. We have simple diffusion. Simple diffusion is just crossing the membrane, diffusion across the membrane. You can see that we have all these particles up here on the top left and none inside the cell. In the middle column, you can see the cell's membrane in the middle here, and we can see the particles slowly diffusing across the membrane. No energy is needed right now. It is simply just kind of making its way through and going from areas of high concentration to areas of low concentration. And in the end, we will have the same amount of particles on the outside as we do on the inside. There's also facilitated diffusion. So simple diffusion was just molecules crossing by themselves. In facilitated diffusion, we actually have membranes, okay, or we have transmembrane proteins. So these integral proteins that go the length of or through the membrane are helping to move particles into the cell. Usually they're going to be larger polar molecules that can't get across the membrane due to the nonpolar tails of the phospholipids. So these are just kind of like corridors, hallways, whatever you want to call them, that allow the particles to get through. Again, no energy is needed. I will also post a video to show how facilitated diffusion works. The last two you didn't need any energy for. With active transport, you do need energy. This is like trying to fight the way against the crowd. If you're walking to your locker from the lunchroom right before lunch and everyone's walking to the lunchroom, it takes a lot of energy to, bob and weave around those people and fight against the crowd. Active transport uses proteins again, protein pumps, and we do need ATP, which you can see down here on the bottom left. We need to give up one of these phosphates, which is kind of like giving, paying the toll at the bridge. We give up some money to allow the sodium in and the potassium out. It costs a little bit of money. And then also, molecules are moving against their concentration gradient. So instead of going from high to low, they're actually going from low to high. Another way we can move things around the cell is with vesicles. Vesicles, whenever we talk about them, I think of Glinda the Good Witch when she floats in on the Wizard of Oz. And it's just kind of a bubble, okay? The bubble is made up of the phospholipid layer around it, and it just moves things. We get vesicles from the rough ER, okay, where we manufacture proteins, and then also to the Golgi apparatus, because the Golgi apparatus is kind of like the UPS postal service of the cell, where it starts to package things and ship them out of the cell. Two places you can see vesicles are when we do endocytosis, which is the internal taking in of substance, entering the cell. Okay, and again, I'll put up the link for endocytosis so you can see the animation. And then we also have exocytosis, which is excreting, things are exiting the cells. Here's two pictures we have for exocytosis. We have constitutive secretion, which goes on constantly. So this would be, the cells like in your nasal passage that are constantly secreting mucus to trap bacteria and dust so that it doesn't get into our bodies. And then on the other side, we have regulated secretion. These are going to be more like your neurotransmitters that once that cell gets a signal, then it's releasing those vesicles to the cell membrane and then releasing them out into the intercellular area. Endocytosis, we have two major ones that we talk about. The first one is phagocytosis, which is the taking in of a solid particle. Okay, you can see how the membrane kind of loops up and eventually it would go all the way around and then it will bring it in. This is called phagocytosis. It is cell eating. Okay, it's solid particles. Next to it in the middle, we have penocytosis. And this is when we bring in a liquid. Liquid is for peno, food is for phagocytosis. And then on the end, we have receptor-mediated endocytosis. This is when we just want to bring in very specific areas. So you can see these little receptors on the end here are trying to grab the straws. Once we have enough, we make the vesicle and bring it inside the cell. As mentioned before, phospholipids can move freely past each other laterally, but they can't move up and down or flip. The plasma membrane is embedded with proteins. That is back in the first cell membrane video. The cholesterol helps stiffen the membrane and make it so that they can't always be moving. And this is because of the fluid mosaic model. I will post links to the other animations. Otherwise, review the video and make sure you know what you need to do for your test.